Hello everyone, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati. And what we were discussing about the different properties of the enzyme in the course Enzyme Science and Technology. And so far what we have discussed about the history of the development of the enzymology. We have also discussed about the naming and as well as the classification of the different enzymes. And then previous two modules, we are discussing about how you can be able to produce the protein or the enzyme in the bulk quantities. In this context, we have discussed about the cloning of the enzyme from the genome. Either you use the genomic library or the cDNA library to select the gene of your interest, or you can use the polymerase chain reaction to amplify the gene of your interest using the site specific primers. Once you have the gene of your interest, then you can digest that with the restriction enzyme, and you can clone that into a suitable vector. Once you got the clone into a suitable vector, then you can transform that into a into the into the host, and that's how you can devise the different strategies to overexpress the protein into the host. So all these processes, what we have discussed so far, are called as upstream events or upstream processes, where you are going to clone the protein, and then you're going to transform the protein, and then you are going to overexpress the protein into the host cells. Uh, Now, what we are going to discuss today is, once you have the protein in the host cells, how you can be able to recover that protein and how you can be able to purify that protein using the different chromatography techniques. So this is what we said, right? We have discussed so far about the different types of upstream events. So in the upstream processes, we are going to do the cloning of the gene. You're going to do the transformations or the entry of the gene into the host, and then you are going to do the overexpression. Once you are going to do the overexpression, you are going to get the overexpressed cells. Okay, these overexpressed cells actually can provide you the protein either into the extracellular media, or it can be intracellular proteins. If it is an intracellular protein, then it has to be the cell has to be broke open so that you can be able to get the the crude product or the crude protein, right, where you are going to have the protein of your interest, and along with that you are going to have the other proteins. From this, you can actually be able to perform the different types of procedures to purify this protein so that you are going to get the final enzyme at the end. So in this process, what you see here is that the first event or the first process is that you are actually going to do the cell disruption. You are going to do a cell disruption so that the material what is present inside the cell is going to be released. And cell disruption can be done by many methods. So in today's lecture, we are going to discuss about the cell disruption method, and then we are also going to discuss about the basics of chromatography. So let's start that. So when we talk about the cell disruption methods are dependent on to the host cells. So you can remember that we were talking about the different types of host cells. We are talking about the prokaryotic cells. We are talking about the plant cells. We are talking about the yeast cells, and we are talking about the animal cells. So the prokaryotic cells are the bacteria where we can actually overexpress the protein, or you can have the eukaryotic choices like the plants, yeast, or animals. So depending on the composition of these host cells, you can be able to devise the different types of disruption methods. So you can have the disruption method which are actually going to withstand, which are going to challenge the physical strength of these cells, or you can actually be able to use the disruption methods which are actually going to destroy the chemical composition of these cells, or you can also try describe try the disruption method, which are actually going to destroy the structure of the cells so that the cells are going to be break open. So depending on this, you can actually have the three different types of method. You can have the physical method where you are going to use the physical parameters. You are going to vary the physical parameter, and that's how it is actually eventually going to lead to the the lysis of the cells. You can actually be able to use the chemical or the enzymatic method, which are where you are going to use the different types of chemicals, and they will actually going to react with the plasma membrane of the cell, and that's how they are actually going to release the content. Or you can use the mechanical method. So you can use the different types of mechanical method, and that mechanical method is also going to disrupt the cells. So let's start first with the physical method. So physical method, these methods play with the physical parameter to damage the cells to release the product. The first is the thermolysis. So thermolysis is a step where you are actually going to use the temperature. So this method is easy, economical, and require no additional specialized equipment. It can be used only if the product is thermostable, which means thermolysis you can do only when the product is thermostable, which means it is not sensitive for the temperature changes. So this method gives a heat shock to kill the organism, and as a result it disturbs the cellular integrity without affecting the product. The effect of heat shock depends on the ionic strength, presence of the chelating agents such as EDTA, and the presence of other proteolytic enzymes. So what happens is that when you are actually going to have a cell, and if suppose you are actually going to increase the temperature, like for example if we heat the cell, right? So what will happen is that it is actually going to expand, right? So it is going to expand to some degree, right? Because you are, when you know that when you are heating that any material, it actually going to expand, right? When it expands, it actually required to synthesize the new lipid molecule, right? So it has to be synthesized the new lipid molecule because now the surface area or the volume of this cell is gone up, right? And that is actually going to be linked to the production of the new lipid molecules, right? And as well as the protein molecules. This means you are eventually going to put a stress onto the cellular production machinery. Now, if you increase the temperature further, so what will happen is that this cell has a capacity until it can be able to provide the new lipid as well as the new protein molecules for the synthesis of the increased surface area of the plasma membrane. And as a result, eventually what will happen is that it is actually going to break, right? It is going to disrupt because it cannot synthesize that much amount of lipids, and as a result, it is going to break open. So apart from that, the another way to explain this is that when you increase the temperature, actually the cellular molecules what is present inside the cell is actually going to push the cell from all the ends, right? Especially like water molecule, right? And they will actually going to create the pressure. So if it is under the high pressure, it is actually going to withstand that pressure if it is actually going to be increase the volume, right? But if it cannot withstand that pressure is going to break the plasma membrane, and that's how the content is going to be released. Then we have the second method, and second method is the osmotic shock. So most of the mammalian cells have a plasma membrane with the active transporters to maintain the osmotic balance. Maintaining an osmotic balance in an active process with the expenditure of the energy. Prolonged exposure to the cell with hypotonic liquids such as water causes osmotic imbalance and ultimately causes the lysis of the cell, right? In this process, due to the inflow of water, cell swells and ultimately burst to release the product. According to the Hoffman's equation, the osmotic pressure is directly proportional to the concentration of the solute and the temperature, and it can be expressed with this particular formula, that is the osmotic pressure is equivalent to RT Ci minus Co, whereas the Ci is the Ci minus Co is the difference between the total solute concentration inside and outside the cell, whereas T is the absolute temperature, which is going to be given in the Kelvin, and R is the gas constant. So if you put all these values, it is actually going to tell you what is the osmotic pressure it is going to cause, and accordingly you can be able to predict when the cell is going to be lysed. So each mammalian cells has a different susceptibility towards the osmotic shock. For example, I have shown you here the red blood cells. So these are the control cells where I have incubated the cell in a isotonic solution such as PBS. Whereas when I am adding the water to this, the cells are actually taking up the water, and that's how you see the cells are actually going to be get lysed. So this is the osmotic lysis. So red blood cell is a very simple example so that to monitor or to actually demonstrate the osmotic lysis. So what you can do is just take the small amount of red blood cells and add 1 drop of water. So when you add the 1 drop of water, what is going to do is the water is going to try to enter into the cell, and as a result the cell is actually going to swell, right? So it's going to swell in size, right? And when it swell in size, it cannot be able to synthesize or provide the additional amount of lipids and protein, and that's how the cell is going to be burst. RBC cell lysis with the lysis with the addition of a tiny drop of blood. Plants or bacterial cells are more resistant towards the osmotic lysis because of the thick cell wall. So bacteria or the plant cells are not susceptible for the water lysis. That's why you have seen that we actually wash the plants, right? We actually wash the plants and we provide them before you cook any kind of vegetables. You actually wash it very thoroughly, and that does not mean that it is actually going to cause any kind of damage to the plant cells. Same is true that plants are also, you are putting the watering the plants also, and that does not cause any kind of lysis of the stem cells or the root cells. Then the third method is called as the sonications. A sonicator generates the ultrasonic waves of the frequency more than 20 kilohertz to cause the cell disruption by the cavitation method. The interaction of the ultrasound with the liquid causes the compression and the decompression very rapidly. The bubble formed in liquid compresses several thousand atmosphere and gives atmospheric shock wave to the cell wall or the plasma membrane to cause the cell lysis. This is what you see here is actually a sonicator where you have the, so this is actually, you see there's a glass actually, so within that the sonicator is being placed, and what you see here is this is the machine through which you are actually going to generate the sonic waves, and this is the probe, and so this probe you have to insert into the vessel where you want to do the sonication, and when you do the sonication it is actually going to generate the bubbles, and these bubbles are actually going to roll over the cell. Okay, so you can imagine that if I have generated a bubble here, what will happen is the bubble is actually going to roll over the cell, and that's how it is actually will go, right? When it roll over the cell, it is actually going to expand the cell because it is actually going to cause a pressure difference, and because of that pressure difference the cell is going to expand, and in this process when it happens very rapidly the cell cannot be able to adjust its volume, and that's how it's going to be lysed. Generation of the ultrasonic waves in the liquid causes rapid change in the temperature and may cause the thermal denaturation. Hence, the ultrasonication medium need to be cool and a long duration should be avoided. So all the sonication, because the sonic waves are very high energy waves, it is actually going to cause the temperature change in the media where you are doing the sonication. So sonication has to be done under the cold conditions, and you are going to put the ice and other kinds of things.
Hi everyone, myself Swayam Prabha, a research scholar at Department of Biosciences and Bioengineering, IIT Guwahati. In this video, we will demonstrate how to sonicate the bacterial pellet, how to lyse the cells, bacterial cells. So we can lyse bacterial cells in multiple ways, like sonication, one of the method. Apart from that, homogenizer we can use. Uh, in this video, we will show you how to use a sonicator and lyse the cells. So after centrifugation, we will get the pellet. Okay. So for this pellet, we have to add lysis buffer of your choice. If you are lysing GST containing protein, so for that you need special buffers or if you are using mannose binding protein containing protein, for that you need special buffer. So it depends on which insert you have and conjugated to uh, what protein. So here this is a histagged protein. In further videos we will show how to purify this protein using nickel NTA column. So for now, after pelleting down, uh, we have to add lysis buffer to cells and suspend, and we have to resuspend the pellet in lysis buffer. So while doing this, make sure that there is no clumps in cell pellet, and always keep on ice. So why we are keeping on ice? Here we have to remember most of the proteins heat sensitive. So to prevent degradation or uh, dysfunctioning of the proteins, we'll use ice as a medium. Like if you keep on ice, at least they will be stable. And also during sonication, we should note that high amount of energy generated. So to dissipate that heat, we will use ice. So I will show you how to operate the sonicator instrument. This is the sonicator. So this is the jacket where we can adjust height of the sonicating sample. So this is once you keep the sample you want to sonicate, you can adjust the height by changing this knob. So actually this is the probe. We are using probe sonicator. Normally for larger amount of samples we use this one. We have small one also. If you are using a pen drop, for that you can use small one. So while sonication is going on, you have to properly close the door. If you are standing there, you have to use headphones. So whatever the sonic waves are coming, it will not affect the ears. Sonic waves are very powerful, and the most affected organism in our body is ears. So it may affect internal organs also. So it is better to keep headphones while sonication is going on.
Then we are going to talk about the chemical and as well as the enzymatic method. So in the chemical and enzymatic method, you are going to use the different types of chemicals, and these chemicals are actually going to react with the cellular machinery, and that's how they are going to cause the cell lysis. So the first is you are going to use the alkali treatments. So alkali treatment, this is harsh but effective chemical treatment to lyse the cells. Alkali treatment causes lipid saponifications which disturb the lipid packing and it affect the cell wall integrity. So lipid means the alkali treatment is you are going to treat it with the sodium hydroxide or other kinds of alkali like KOH and other alkali. So what will happen is that it is actually going to it is actually going to dissolve the lipid molecules, and that's how it is actually going to disturb the lipid packaging, and it is going to affect the cell wall integrity. Then the second what you're going to use is you're going to use the detergents. Adding a detergent solution to the cell causes solubilization of lipid to form the micelles. The effect of detergent on the cell wall increases linearly with the concentration. The detergent concentration, which causes abrupt change in the lipid solubility and form micelle, is known as the critical micelle concentration or CMC. For example, the detergents are the SDS, sodium dodecyl sulfate, CTAB, Tritonax 100, saponin, and gitonin. So what will happen is that if you are taking a cell and if you are adding the detergent, right? If you are adding the detergent, so detergent is what? Detergent is a amphipathic molecule. So it is actually going to have the hydrophobic surfaces. So what will happen is that these lipid molecules are actually going to be get dissolved into that, and as a result the cell is going to be devoid of the lipid, and when you don't have the lipid it is actually going to disrupt the plasma membrane. The some slightly different process where also you can use the detergent is called as the permeabilization. So if you use a very high concentration of detergent, it is actually going to remove the large quantity of lipids from the plasma membrane, and that's how it is actually going to solubilize the cell. So there will be no plasma membrane, so all the cytosolic content is going to be released. Whereas if you remove the lipids on a partial matter, right? If you remove the lipids, like for example, this lipid, this lipid, this lipid, this lipid, you're not going to remove all the lipid molecule. Then this process is called as the permeabilization. So permeabilization is actually not going to remove the all the lipid molecules, but it is actually going to make the tiny holes, and these holes can be used for delivering the content into the cell. A partial cell wall disruption or the permeabilization is achieved by the organic solvent such as toluene. The organic solvent is absorbed by the cell wall, resulting in its swelling and ultimate rupture. But at low concentration, the organic solvent permeabilizes the cell wall without disturbing the cellular integrity. This process allows to use cell as a reaction vessel to catalyze the reaction and get desired product. So permeabilization has very good advantage, because it actually going to give you, it actually going to make the cell as the vessel, right? So you can convert the cell which is into a reaction vessel, right? So you can imagine that if I'm making it as a reaction vessel, I can actually be used that for catalyzing the reaction, because all these tiny pores will not allow the release of the content. So all the content is there, but this cell is dead actually. And I can put, like, for example, if I want to convert the glucose into glucose 6 phosphate, right? I can do that because it actually has the hexokinase, so it actually can convert the glucose into glucose 6 phosphate. So what I can do is I can just formalize the cells and immobilize to some surface, right? And in that case, this will work as a bag of enzymes actually. So I can do all the chemical reactions, all the sorts of modifications, and so on. Then you have the enzymatic method. Okay, so enzymatic methods are specific, gentle, and most effective but costly. Lysozyme is commercially available to treat the bacteria to release the intracellular product. In addition to lysozyme, there are three other types of bacteriolytic enzymes like the glycosidases, acetylmuraminidase, L-alanine amidase, and the endobacterase. Food proteases are also found to be bacteriolytic. Each cell lysis require a mixture of different enzymes such as glucanases, proteases, manases, or chitinases. Plant cell can be lysed by the cellulases and the pectinases. In most of the enzyme-mediated cell lysis method, the rupture of the cell wall depends on the osmotic pressure of the external media. In few cases, the enzymatic digestion is performed to remove the external cell wall, and then in the second step, the protoplast is disrupted by the gentle addition. So the enzymatic digestion is being used sometime to even remove the outer covering. For example, you take the plant cell. So what will happen is in the plant cell you have a cell wall, and within this you are actually going to have the plasma membrane. So what you're going to do is you just treat it with the cellulase. So what will happen is cellulase is actually going to chew up all the cellulose molecules that is present in the cell wall, and that's how it is actually going to generate a plant cell without the cell wall. Now this cell is actually sensitive for the osmotic damage. So what you can do is then you can just add the water, and it is actually going to break open the cell. Because as I told you, right, the plants are not sensitive for the osmotic damages, but that is only because there is a cell wall present, right? But if you treat it with the cellulase enzyme or pectinase enzyme, it is actually going to remove the cell wall, and that's how you're going to have the plant cell without cell wall. And then if you add the water, it is going to be susceptible for the osmotic lysis, and that's how you can be able to release the content of the cell. Now let's talk about the mechanical method. So mechanical method are going to be the method in which you are actually going to use the some kind of machines. So it in the simplest mechanics used in the laboratory is the Waring blenders or the pestle mortar. So Waring blender is nothing but the mixer grinder actually. Okay, I'm sure you might have used the mixer grinder for making the chutney, right, in your home, right? So when you prepare the chutney is nothing but the cellular homogenate, right? It's a homogenate actually, because where you are actually taking the different types of constituents, like you're taking the corianders, you're taking the onions, garlic, and all that, and then you are running the mixer grinder, and that's how it is actually going to crush all the cells, and it is giving you a paste. That paste is nothing but the cellular homogenate. Okay, so it actually contains all the different types of enzyme and so on. Pestle mortar is also the same. I'm sure you might have seen the pestle mortar. It's where you have a bowl and you also have a rod, right? So that rod is actually going to be used for grinding the cell, grinding the substance. But these are mechanical method. These are the manually mechanical method. Whereas you can actually be able to use some more effective method where you can be able to run the motors, and that's how the it is going to be more and more effective. Both are effective towards the animal and plant tissue as well as the filamentous organism. So you can use the Waring blender or pestle mortar, which are effective for the animal and plant cells, but they are not good for the hard tissues, right? And they are also not good if you are trying to do this at an industrial scale, because you cannot do the industrial scale mixer grinders, right? So in those cases, you have to use the bid mill homogenizer or the high pressure homogenizations. So in a bid wheel homogenizers, disruptions, what you are going to have is you're going to have the bead mill homogenizers, right? So the bead mill homogenizers, either it can be or a horizontal model or it can be a vertical model, which means it is either going to be in this direction where you're going to have the shaft on this side, or it can be actually this way so that you can have a shaft on this side. Either of these consist of a grinding cylinder with a central shaft fitted with a number of impellers. So this is the central shaft what you have, and then these are the impellers. So they run in a clockwise direction or the anti-clock directions, which can move in the clockwise or the anti-clock direction with the help of a motor. So this is actually being connected to a motor, right? So this is being connected to a motor, and that motor can run these impellers into the positive in the clockwise direction or the anti-clockwise directions. The grinder cylinder is filled with the bead made up of the glass, alumina, titanium carbide, zirconium oxide, or the zirconium silicate. There is an inlet to supply the cell suspension and an outlet to collect the sample after the process. When the bead mill runs, cell experiences the shear stress between the moving beads. So what will happen is you are actually going to add the beads, and so this is the the bead wheel homogenizers, and in this you have a central shaft. This shaft is connected to the motor, and on this shaft you have the impeller. So when this runs, it and you're also going to add the beads, right? So when these beads actually moves, so what will happen is that you can imagine that if these two beads are moving, the cell is actually passing through to these beads. So cell is passing through these tiny pores which are being formed between these beads. So as a result, the cell is actually going to experience a shear stress, which means these two beads are actually moving and the cell is hitting with these beads, and it is actually passed through. So it actually has to squeeze, and then only it can actually be passed through. But in that process, it is actually going to experience a very significant change in pressure, because there is a very high shear stress, and as a result, the content is going to be released. Uh, then we have the high pressure homogenizers. High pressure homogenizers consist of a high pressure positive displacement pump connected to the adjustable discharge wall with a restricted opening. The cell suspension is sent into the homogenizer through a small homogenizing wall at a very high pressure, like 200 to 1000 atmospheric pressure. The cell passing through a small wall experiencing the shear stress developed within the liquid, and as a result it get disrupted. So what you're going to do is you're going to add the cell, and then you have actually the this is the high pressure homogenizer, where this valve is actually can move in the up and down direction. So when it goes up, all the cells are actually passing through this tiny pore, and it is actually coming out, right? And when it is coming down, all these cells are going again back. So when these cells are passing through these small pores, they are experiencing a shear stress. And the pressure difference, what you see, is from 200 to 1000 atmospheric pressure. This means the pressure is going to be very high. So when it happens with a very high pressure, all the cells are actually going to be released, going to be released, and they are actually going to release their product. And then this product can be recovered from the another opening. So this is all about the cell disruption method, right? So now once you have done the cell disruption, you are going to have the crude product. What is there in this crude product? The crude product is actually going to have the enzyme, what you are going to be purified, and then it is actually going to have the protein from the host, right? So it's going to have a mixture of the two species, one the proteins from the host and the enzyme what you are overexpressing in a large quantities. Now our objective is that we want to purify this particular enzyme, so we want to isolate this enzyme, and that's how we are actually going to do a technique which is called as the chromatography. So you're going to purify this with the help of a technique which is called as chromatography. So with this, I would like to conclude my lecture here. Thank you.